So the first module of the EEG Basics lecture is on underlying brain processes of the EEG. So fundamentally, whatever your BCI is doing, at the end of the day, you have to operate on some effects that are actually observable from your sensors, right? Observable effects of brain activity. There's basically, as I said, two big areas, and one is all this related things, all the effects that are related to blood flow and blood oxygen level and so on, which fMRI and fNIRS do. And then there's everything else, and that everything else is primarily electrical phenomena, at least today. There's also chemical ones, but that's not yet being done today. So electrical processes of, and that's field effects of neurofiring processes. So there's lots of neurons in there, right? 100 billion or so. And they fire and they have subthreshold activity as well. That's what BCIs, EEG-based and otherwise, usually operate on. And for the easily wearable, non-invasive measures like EEG and EEG, or others like MEG and ECOG and so on, all of these have one thing in common, and this is that they can only pick up large-scale or coarse-grained electrical processes or processes of large neural populations. And so consequently, what you can ultimately reason about and work with are things that affect lots of your brain dynamics. And if you look back at the examples of applications, you see these are usually, large-scale operator state changes or person cognitive state changes like workload and so on. It's pretty clear that that is exactly in that category. So it might be something like 50,000 neurons firing at the same time, or a centimeter-sized patch of the brain doing something in synchrony. And I'll show you how that can happen. So the primary contributor to EEG are the pyramidal cells. It's these greenish here, which are sort of aligned in such a way that they're orthogonal to the surface of the cortex, the folded matter. You see here's the surface, here are some of these cells. And the important part is really that they're aligned, because when they fire or have subthreshold activity, that activity adds up instead of canceling out or something like that. And that's precisely why you can measure it, say, from up there with other noise sources interfering and so on. It's not about whether there is any field measurable outside the brain, but it's about whether it reaches above your noise floor. Sensor noise, thermal noise, and all that. That determines what you can ultimately measure, and that gives rise to the minimum number of neurons that you have to have doing the same thing at the same time. It also only happens when they fire at the same time, right? It doesn't matter. If it all goes willy-nilly, you're not measuring anything. And so the question now is, why would, say, 50,000 neurons fire at the same time? And there's only that many kind of situations that give rise to that. One is very clear. If you have an external event, say a flash of light or so, that triggers a cascade of change through your brain, like a ripple, if you will. And that gives then rise to kind of co-activated firing of many neurons. And lo and behold, that's something that actually gives rise to working BCIs. There's an event, like you saw something or you heard something, and the BCI operates on any EEG activity following that as a consequence of this synchronized activity. The same also works for internally generated events. So if you have a sudden aha moment or so and realize something, that gives rise to a lot of change in your brain, lots of dynamics that are happening at the same time. And so you might be able to pick up a signal out there, like a peak or so. The only thing is, in that case, you may not actually know when to look because you didn't observe the aha itself. And there's something that's fundamentally kind of different than that, and that is neuropopulations that happen to be synchronized because they synchronized themselves with each other into a kind of a steady state firing pattern. And that can be because there's a mechanism in the brain that gives rise to this kind of firing synchronization, or, ultimately there has to be a mechanism. And one of the biggest phenomena in EEG that exhibits that are oscillations, which are, for example, the so-called alpha waves, which is many neurons firing at about 10 hertz, and they're all in sync. And it mostly doesn't happen when the neurons do some meaningful task, because if they're doing meaningful tasks, it goes all over the place in time. It happens when the neurons are not engaged in any task, when they're idle. Neurons become idle, and in some areas of the brain, there's mechanisms that give rise to co-activated firing of these populations then. And that gives you a signal that you can pick up out there, so to speak, with your EEG, which is somewhat counterintuitive. Why do you have more activity if there's less stuff happening in the brain? Well, that's the reason, because it's kind of synchronization then. And so that gives rise to the two big categories of EEG phenomena. There's so-called event-related potentials, which is these two guys, and then there's oscillatory processes, which is the other one, synchronized firing. These are two dominant phenomena. There's certainly others. There's things that are not really strictly oscillations and so on, but there exists way more technology that deals with this than anything else at this point. So I already hinted at that. So there is the question of what you can detect with EEG. Which of these phenomena can you detect? As I already indicated, you may not directly observe the root cause of what you see. So you don't observe the aha moment itself, the first neuron that fired or so. You may observe the cascade of changes that comes with that. Or you might see changes in cortex in the neurons that is actually modulated by chemical changes in deep structures of your brain, such as more dopamine or things like that. So you never observe with your electrodes these chemical changes, but you see cortical correlates of that. Fundamentally, when we're talking about synchrony, there's one thing, and that is if you're looking at, say, a broad patch of neurons that are not directly connected by fiber tracts. These neurons are rather unlikely to fire together unless they are sort of being prompted to fire by the same input or so from an external event. So because they have no mechanism to synchronize themselves, any propagation from one neuron to the other just takes way too long to give you co-activated firing. So it's much more likely that spatially compact populations of neurons actually fire together, like a centimeter-sized population of neurons. They're close enough and propagation delays are low enough that they can all go up at the same time, in a sense. And that's, in fact, exactly what we see in EEG. I'll show you a few pictures of that later, that it's kind of compact generators. There's also cases where you have compact generators. They fire at the same time. You should be able to observe it, but you see nothing. And the reason is because the signals can sometimes cancel each other out. So there's one case that's the amygdala, which is sort of a spherical structure deep in the brain, which is related to fear and depression and things like that. It's sort of a closed field. So the neurons are sort of at the outline of the sphere. And when they activate, the signals exactly cancel each other out. So there's nothing that you observe in the EEG. But you may see related changes that are modulated by that. And that is basically the end of this module.